Development of the eyes. The following two figures show major parts of the eyes. These structures develop from several embryonic layers. The lens and part of the cornea are derived from surface ectoderm and mesoderm. The neural layer of the retina, the posterior layers of the iris, the optic nerve, the ciliary body, and the iris are formed from neuroectoderm. The choroidal layer, outer fibrous sclera, and corneal endothelium develop from mesenchyme that is believed to be of neural crest origin. The vascular endothelium of the choroid is derived from endoderm. The anterior chamber develops from a slit-like space in mesenchyme that is located between the developing lens and cornea, and the posterior chamber also develops from a similar space that is located between the developing lens and iris. The vitreous humor, the fluid component of the vitreous body, is derived from mesenchymal cells with neural crest origins. This 22-day-old embryo shows fusion of the neural folds with open and posterior anterior neuropores. The earliest indication of eye development is the appearance of the optic grooves in the forebrain region of the embryo. Let us now look at a transverse section of the embryo at this level to show the fate of the optic grooves. Observe how the optic grooves evaginate and form hollow diverticula, projecting laterally into the periocular mesenchyme. The optic diverticula dilate to form the optic vesicles, which are connected by the optic stalk, primordium of the optic nerve, to the developing brain. At day 24, the vesicles come into contact with the surface ectoderm. The ectoderm adjacent to the optic vesicles thickens to form the lens placodes, which invaginate and sink in a lens pit below the surface of the ectoderm. Inductive messages pass from the vesicles to the placodes, resulting in the formation of the lens primordium and later the lens vesicles. As the lens develops, the optic vesicles invaginate to form double-walled optic cups. Hyaloid vessels are formed from mesenchyme in linear grooves, known as retinal fissures, along the ventral surface of the optic cups and optic stalks. The hyaloid fissures close by the end of the 7th week. The hyaloid artery in the retinal fissure supplies the inner layer of the optic cup and the lens vesicle. Distal parts of the hyaloid arteries degenerate, and the proximal part persists as the central artery of the retina.